And welcome back to Geology 309, Week 7, Part 2. In this recording, we are going to be talking about Superphylum Lophophorata. This includes the two phyla within, Brachiopoda and Bryozoa. We left off last time talking about coelomates a little bit. We were talking about this larger group that includes most of the organisms that we're going to be talking about. A coelomate is an organism that has an internal fluid-filled body cavity, or a coelom. This also includes us. They have differentiated organs, so something other than just a large body cavity to digest. They have a specific digestive and excretory tract. They have specialized reproductive organs and a musculonervous system and so on. Okay. So most of the organisms on the planet are coelomates. In other words, it includes Cnidaria, not Porifera or Archaeocyatha, and pretty much everything else that we're going to be discussing. So here we again look at our phylogenetic tree that we started looking at last time. We discussed poriferans already, and now we are going to be looking at coelomates. Remember, this includes cnidarians, but once we get into this subdivision of coelomates that have bilateral symmetry, we get into these two groups, protostomes and deuterostomes. So let's look at those two groups a little bit more. So what is a protorostome and what is a deuterostome? These are two different major groups of coelomates, and we differentiate them based on embryological development, actually. So this is something that we know based on modern-day organisms, and then we can apply it to fossil ones. So we're talking about what's happening at the very earliest stages of embryological development. So before these organisms are even born, when there's just a couple of cells starting to form. So remember, an embryo starts as like a clump or a ball of cells. But we as living organisms have openings in which we eat, we excrete, we breathe, we do all sorts of things. Which body opening is forming first? And the way that this happens is that ball of cells starts to get a little indentation as it continues to reproduce more and more cells. Some of these indentations will form the mouth. Some will actually form, well, the anus, the butt. And that's how we differentiate proterostomes and deuterostomes. A proterostome is an organism where, as an embryo developing, the first gastrulation or indentation of cells forms what will become the mouth. This includes the lophophorates that we're about to talk about next. So it includes brachiopods and bryozoans. It also includes all arthropods, so bugs, crabs, crustaceans, and annelids, worms. It also includes all of our mollusks, so clams, gastropods, and cephalopods. Deuterostomes, however, are an organism where, as that embryo develops, the first gastrulation forms the anus. This includes phylum Echinodermata and chordates, actually. So we, as chordates, develop butt first. All the other organisms, except echinoderms, form mouth first. It's good to have some pride in yourself. This is what we mean by gastrulation. So if we look at stage A, looking at either a proterostome or a deuterostome, doesn't matter, you see this ball of cells. There's about 8 cells at this point. Now, during part B, we form the coelom, that internal body cavity, and that forms as more and more cells start to reproduce. It folds in on itself. Now, that inward fold either becomes the mouth eventually in the form of proterostomes, or that blastopore opening forms the anus, which is what we do and echinoderms do. Okay, so the blastopore, that first indentation of cells, either forms the mouth in proterostomes or the anus in deuterostomes. So we're going to be talking about both, but most of the organisms we're going to be talking about over the next few weeks are proterostomes, and we'll get to the ass-first organisms a little bit later. Phylum Brachiopoda and Bryozoa both fall under Superphylum Lophophorata, because both of them possess a lophophore, which is a specialized filter feeding apparatus that's somewhat fan-shaped. If you look at the inside of a brachiopod valve set, you'll see in many cases a coiled or spiraled feather-like structure that they use for filter feeding. So they open up their valves, they extend out their brachidium with the lophophore, and they filter feed that way too. Bryozoans, though much smaller, also have a very similar structure. So thus we combine both of these under the umbrella of Superphyla Lophophorata. There are some other characteristics or situations that brachiopods and bryozoans share. Both groups are relatively minor in their diversity today, but were incredibly abundant during the Paleozoic, especially in the Ordovician. Today, bryozoans are present in marine and fresh waters around the world, but the diversity is much lower. Similarly, with brachiopods, if you were to go collecting shells on the beach back in the Ordovician period, you'd be collecting brachiopod shells, and today it would be bivalve mollusks. So even though brachiopods look like bivalves, they're entirely unrelated. Similarly, even though bryozoans kind of look like coral sometimes, they are again unrelated. Let's take a look at the anatomy of a brachiopod set of valves or shells. There's two main shells. We have the pedicular valve, which is the upper one, and the brachial valve, which is lower. The pedicular valve is called such because it has this opening known as the foramen in the back and lower portion of the pedicular valve that the pedicle, which is a fleshy foot-like stalk, would extend out and from and anchor the brachiopod to the substrate. They then open up the pedicular and brachial valves and extend out their lophophore to feed. The larval curvature that we see on the brachial valve, so in our life reconstruction, it would be this area down here. Inside view, though, this structure here is known as the umbo, very similar to something that bivalves have as well. You can see radiating growth lines from the brachiopod also on the pedicular and brachial valve. In between the foramen and the umbo, we have this triangular-shaped depression known as the deltidium, and this assists in muscle attachments that hold these two valves together. So there are about 4,500 or more genera of brachiopods described, but not many of these include living examples. They are bivalved or bivalvate. In the case that we mean that they have two mineralized shells, but that doesn't mean that they are a bivalve mollusk. It's a similar terminology to saying an organism is amphibious, meaning it lives in land or water, versus an amphibian, which is a type of organism. So in this case, they are bivalved, but they are not bivalves. They're not bivalve mollusks, but they have a bivalvate condition. They are solitary, unlike some bivalve mollusks, which will grow in groups or clusters. Brachiopods may grow close to one another, but they don't actually attach to one another. They're solitary. They are also sessile, which means that they stay put. They don't get up and swim around or usually get up and move around very much at all. Most of them are epifaunal, living attached at the sediment-water interface, though one group is actually infaunal and will burrow down. Most, again, attached at the substrate, though some were free living on sandy or muddy bottoms and would attach onto other structures, such as branches of plants or other types of organisms. Brachiopods are also strictly marine, which also separates them in lifestyle from bivalve mollusks, which can be found in marine, brackish, or fresh water. Bivalves are strictly marine. They also have generally a very slow metabolic rate. This means that they can live in very deep water environments with relatively low oxygen settings, and most of what they're filter feeding is any organic material that's suspended in water. Again, to focus a bit on the morphology, we find that brachiopod valves are either made of calcium carbonate or chitin, which is the same type of material that your fingernails are made out of, or insect carapaces. So it's an organophosphate in some of our different versions of brachiopods. They are heterovalvate, and now this means that the two valves, the pedicular and brachial valve, are dissimilar in shape. Contrast this with many types of bivalve mollusks where the two shells are similar to each other but just symmetrically different, like they're mirror images of each other. In this case, the brachiopods, the two valves are very dissimilar. However, each valve has a plane of symmetry, and it's bilateral, perpendicular to the hinge where the two valves are attaching. Again, if you look at a clamshell, the two shells may look similar to each other or symmetrical to each other, but the shell itself is asymmetrical. The opposite is true for brachiopods. The two shells are dissimilar, but each shell has a plane of symmetry. Again, our attachment structure, the pedicle, exits out of the brachiopod's foramen through the pedicular valve. And the shells open and close by two muscles, adductor and diductor muscles. Adductor muscles close, diductor muscles help open. So similar to our bivalve mollusks, which also use muscles to close their shells, brachiopods use a muscle to open and a muscle to close. So they actually have distinctive muscle scars. And by scar, I don't mean an injury, but an attachment point on the inside of the shell. On a living brachiopod, the shell is covered by an organic material called a periostracum. This rarely preserves in the fossil record, though we do have obviously modern specimens that still possess it. Brachiopods also display homeomorphy or convergence in shapes in different groups. So we'll look at a few different types of brachiopods that look very, very similar to one another, but those similarities in shape are actually convergent. They didn't derive them from a common ancestor. If we look at the distribution and classification of brachiopods, we can see that they got a very good, strong start in the Ordovician. We have the width of the bars here looking at numbers of genre. So we have lots in the Ordovician, and then we lose some at the end of the Ordovician. We see a big bounce back in the Devonian, another bounce in the Carboniferous, quite a lot in the Permian, and after the Permo-Triassic extinction, notice brachiopods have not been as abundant as they have since then. We approach to a number of taxa a little bit more than what we had in the Cambrian throughout most of the Mesozoic and Cenozoic, but we don't see anything like what we had in the Paleozoic itself. So if we look at some of the kind of evolutionary flows of different types of brachiopods, the orthids are among the most primitive. And from the orthids, we think we're seeing diversifications into groups such as the strophomenids, the spiriferids, and the pentamerids, which then diversify into the rhynconellids, the atripids, and ultimately the terebratulids. So we're going to go through these different groups and point out some characteristics that help us identify them. There are two main classes of Brachiopoda, Inarticulata and Articulata. We'll start with the inarticulates, and this is really a fascinating group. The valves in this case are unhinged, and what that means is the two shells are held together by soft tissue, like a ligament. When this organism dies and that soft tissue decomposes, the two shells usually fall apart, hence Inarticulata. One of the most abundant orders within class Inarticulata is Lingulida. Lingulids are around from the Lower Cambrian, and we still have them alive today. In fact, here you're looking at a Cambrian lingulid, and the image here obviously is a modern one. They have changed very, very little in that time. This is also our only infaunal brachiopod. That pedicle is very, very long and extends way down into the sediment, allowing the lingulid to pull itself down into the sediment or reach up when it wants to filter feed. Their shells are usually phosphatic, and we'll see that most of our articulate brachiopods actually have a calcium carbonate shell. Another inarticulate group, though, that are quite abundant are the Acrotratidas. These are oval or circular shaped and also epifaunal, so they don't have the long pedicle to anchor them way down in the sediment. Otherwise, they do have some similarities in appearance to lingulids. And these are also still alive today and also go back to the Cambrian. There's a lesser known inarticulate known as the Orbiculoideas, and these are exclusive to the Pennsylvanian. They only have one shell. They only have one valve. Again, it's phosphatic, and they're very, very small. And they have kind of a little cap or slightly curved cone shape to their valves. Class Articulata is much more diverse and shows a lot more disparity throughout the Paleozoic, even into today. These are much more complex, and the valves in this case are actually hinged. So there's a specific structure to the valves that allows them to lock together like a hinge. This means when they decompose, the soft tissue decomposes, the two valves are still adhered to each other. So you usually find them together. And again, the calcite valves here are different than the phosphatic ones that we typically see in Inarticulata. There are 7 main orders of articulate brachiopods. There's one significant suborder that we'll talk about, and lots of other minor orders that we'll discuss to some degree or another. We'll start with some of the more primitive articulate brachiopods, order Orthida, the orthids. These are around from the lower Cambrian through the upper Permian, so they really span the entire Paleozoic era. They have a biconvex shell that is finely costate or ribbed. So the biconvex shell, you'll see that both valves are different from each other, but you can easily draw a line of symmetry through the two valves. The costae, or little ribs, are what are growing outwards. Okay, those aren't growth lines. I'll do the growth lines in yellow. That would be a growth line. So if they are radiating outward, lines radiating out, those are costae or ribs. And if they're circular around the shell, like you see here, those would be growth lines. So orthids have that biconvex shell where the two valves kind of cup into each other, and they are finely costate. They also have what we call a strophic hinge, and it's very short. So in blue, here would be the hinge line, and it's a nice straight line, but it's very short. It doesn't widen the shell out too much. So if you're looking at a brachiopod, you notice that both valves are kind of biconvex. You see that the valves have very fine costae on them, and it has a short strophic hinge. You're probably looking at an orthid. And as far as the age of it, well, it could be anywhere in the Paleozoic. Another order would be the Atripids, which look a little bit like the orthids at first appearance. These are around from the middle Ordovician through the upper Devonian. Inside of the Atripids, and occasionally this does fossilize and mineralize, we have a spiral lophophore. So you'll see these kind of coil spring-like structures. Now, the purpose of this is it allowed the lophophore to wrap around a longer object, which means that's a larger filter feeding apparatus. They also have a very short hinge, but in this case, it's astrophic, meaning that the hinge line is curved and short. It's not straight like the orthids. So at first glance, these may look similar to orthids, but if you really start looking at their characteristics more, they're quite different. Order Strophaminida is a very well-known order of brachiopods. These are around from the Ordovician through the Triassic. They also have a biconvex, in this case concavo-convex shell, where the two shells cup into each other, and the overall shell is very D-shaped. And part of that is because it has this incredibly long strophic hinge. So these are relatively flat brachiopods because both valves cup inward on each other. If you look at these in cross-section, you've got your pedicular valve, and then inside of it essentially is your brachial valve, like that in cross-section. They also have very little or no apparent sulcus. A sulcus is occasionally on a brachiopod. You'll see a depression going down the center, parallel more or less to the plane of symmetry. But we don't really have much of that in the case of the strophomenids. Within Strophomenida, we do have a suborder, and that is suborder Productida. So these are a type of strophomenid. Productids are an incredibly diverse group of brachiopods, very disparate, and they're exclusive to the later part of the Paleozoic for the most part. They're highly derived, concavo-convex shells, just like the other strophomenids. In this case, on the convex part of the brachial valve, they have lots of spines. So they would use these spines to either wrap around structures like twigs that might have been in the water, or they anchor themselves into the sediment. In other words, they're really getting rid of the pedicle. They still have a pedicular valve and a brachial valve, but they get rid of the pedicle. They use spines to anchor themselves in, so the pedicle is greatly reduced. In many of the fossil specimens of the productids, like Linopricductus, for example, you're not going to see much of the spines because they're needle thin, calcium carbonate, and they end up breaking off during fossilization. But on some specimens like this one, you'll actually feel the little nubs where those spines used to attach. These are exclusive to the Mississippian and Permian, so Mississippian, Pennsylvanian, and Permian. They're around in the very late portion of the Paleozoic. And if you think about what's going on in the world at that time, it makes sense. This is when we're starting to see swampier conditions, so attaching to a hard substrate is going to be difficult. So instead, they end up just burrowing themselves into the mud with their long spines. Another order of articulate brachiopods that are quite abundant in Wisconsin are the pentamerids. Pentamerids look like a slightly elongated brachiopod that's been clefted with a hatchet or something. They've got this little cleft at the hinge themselves. So they're from the Middle Cambrian through the upper portion of the Devonian. Again, a very short hinge that is astrophic. And the cleft often appears in this upper pedicular valve. So pentamerida, pent refers to 5, and that's because this almost looks like there's 5 different lobes of these brachiopods because of the cleft that appears at the back towards the hinge line on the pedicular valve. So they're actually widening open a pedicular opening a little bit more. When you usually find one pentamerid, you find a lot. They lived in these clusters. Again, they're solitary, but they're growing all next to each other in very sandy facies. Order Spiriferida is one of the best studied groups of brachiopods. We talked about the strophomenids having a very small or absent sulcus. These have a very deep sulcus. Spiriferids are around from the middle of the Ordovician through the Jurassic period, and they are highly variable. As you can see, just among the few specimens we have on the screen, they come in a wide variety of shapes and sizes. They also have that spiral lophophore like the atripids, and a very well-developed sulcus. One of the more common features we see with spiriferids, though, is a very long strophic hinge producing almost wing-like structures. So again, as we look at our later spiriferids in the latter part of the Paleozoic, just like the productids are producing long spines to anchor themselves into these muddy environments, by growing a long, wide strophic hinge, that allows for a very long, coiled brachydium of the lophophore. Okay, so this increases the size and surface area of the filter feeding structure. Order Rhynconellids, or Rhynconellida, are kind of an interesting group. They're from the Middle Ordovician and actually still around today. They have a biconvex shell, so they form almost a perfect little ball. They also have a very small astrophic hinge, as you can see up here. They have a deep sulcus, but they also have what's called a zigzag commissure. The commissure is how the two valves fit together. So in this case, the zigzag commissure looks like this. So that's the line upon which the two valves are enclosed. So when they open up the valves, it almost looks like they have teeth. But again, remember, they're not biting at anything. They're opening their valves to extend out a filter feeding structure. They also have a very, very small foramen because their pedicle is very, very tiny. Order Terebratulida also involved modern specimens. Terebratulids have been around since the Lower Devonian to today. They're still around. These are sometimes called lamp shells. And you can see they are astrophed. So here we're looking at the brachial valve, and here's the pedicular valve, and there's its astrophic hinge. Very large foramen right there at the beak where the two valves are meeting. And you can see some examples, in this case, of living terebratulids in their natural habitat. So when did we have all of these different groups? Well, again, lingulids have been around forever. Strophomenids really kind of span just about the entire Paleozoic into the Triassic a bit. The orthids cover most of the Paleozoic. Pentamerids are a little bit more in the front half of the earlier half of the Paleozoic. Rhynconellids and terebratulids have a very long history as well, and then the Spiriferids also covering most of the Paleozoic. So it's really, the Paleozoic is a time for brachiopods. And we have 1, 2, 3, 4, 5, 6, 7, 8 different groups present right here. We only have 2 or 3 really that are alive today. Well, what about our other types of lophophorates, the bryozoans? Bryozoans are all colonial. So we're going to see some similarities with bryozoans to corals, just as when we talked about brachiopods, we were looking at similarities and differences with bivalve mollusks. So the thing about lophophorates is they're almost mimics of some of these other phyla. So these are a lophophorate. They do have a lophophore. In this case, it's a tentacled, kind of ciliated, extendable filter feeding structure. They reproduce sexually and they reproduce asexually by budding or kind of cloning themselves. So almost sponge-like in that way. The skeleton of a bryozoan colony, what we typically find in the fossil record, we call a zoecium. And these are made of either calcite or chitin. They're all colonial, and if the skeleton is the zoecium, the individual animal is called a zooid. So think about this in terms of, with a coral, the individual animal is called a polyp. In this case, it's a zooid. A colony of zooids is known as a zoaria, and they live inside of a zoecium. Bryozoans form from a wide variety of different growing strategies, such as branching. They can be columnar, or they can also have encrusting zoaria, encrusting colonies. There's about 4,000 living species of bryozoans. Most of them are marine, but we do have some freshwater taxa. There's about 15,000 extinct species, so these used to be much more diverse than they are today. The first bryozoans appear in the fossil record around the Ordovician, and again, we still have them alive today. And they're major contributors to reef frameworks, especially in the upper portion of the Ordovician. That's when we see a lot of our bryozoans making up significant portions of reefs. There are 3 classes of bryozoans and 7 major orders. We describe them and study them usually in thin section under a microscope because they are rather small, and we use that to determine the size and shape of the zooid and the zoaria. They don't have a very rapid evolutionary history. Again, probably part of that is due to the asexual budding reproduction. But they do have very high diversity. They just haven't changed a lot very quickly throughout the Phanerozoic. So let's look at some like 4 of the Paleozoic orders of bryozoans. The first order, which we see from the Ordovician through the Triassic, is order Terebratulida. This word translates into changing mouth, and by mouth we're looking at the opening or the aperture in which the individual zooids would extend their filter feeding apparati out. These typically form branching, encrustive, or sometimes just large, massive structures of bryozoans. They have a very small aperture for a very small lophophore. However, if you look at the zoecium in cross-section, they're partitioned, so they have little walls separating them off, hence why we use the term changing mouth, because as you look through the zoecium, there's a lot of changes from one part of the colony to the other. We see a lot of these in our Ordovician rocks, so Terebratulida and bryozoans are not uncommon in some of our local rocks around this part of Wisconsin. We do lose a lot of Terebratulida bryozoans along the Ordovician-Silurian boundary, so these are among the fallout from the Ordovician extinction. A couple of them have survived through the Permo-Triassic extinction, but the last of the Terebratulida bryozoans died out at the end of the Triassic period. We can see changes in the growth from juvenile to adult. So we see actually quite a lot of ontogenetic variability in this particular group of bryozoans. Our next order of bryozoans is Cryptostromata, which means hidden mouth. And the reason for this has to do with the external structure of the colony. These have been around from the Ordovician through the Permian, so they span almost the entire Paleozoic. The true aperture is much smaller than what it appears when you first look at it. When you look at these under a microscope, you'll see that the main opening of the aperture, what looks like the opening of the aperture, is actually a vestibule or like a diamond-shaped depression that we see here. Let me see if I can highlight that a little better. There we go. So that's the vestibule, and then there's the actual aperture inside. If you cut these in half, you'll see a tube-like zoecium with these little partitions. These are abundant in our upper Ordovician rocks. That appears to be when they really started out. They had a huge diversification event in the Ordovician, except we do lose a lot of them then at the end of the Ordovician. So some of these are only present in the late portion of the Ordovician, but many do continue to persist until the Permian. Our third order is really kind of a famous one, order Fenestrata, the fenestrate bryozoans. These are around from the early Ordovician through the Permian, but they are especially abundant in the Mississippian. In fact, these are one of these taxa, Archimedes, is the only bryozoan index fossil. They make these very distinct lacy colonies, and it's actually in the crossbars of the lace that we have our individual apertures for each zooid. Going down the center of the colony, though, we have this screw-like stalk on which the fronds of the Fenestrata actually attach. And what this does is it produces a spiral basket, allowing that zoarium to act as a funnel filter. So as kind of mucky, organic-rich water is flowing through that basket, it slows down as it goes through those small openings, and this allows the bryozoan colony to filter feed off as much organic material as they can get. So when you find bryozoan fossils that are fenestrate, there's two different parts to look at. There's the central screw axis, the stalk, and then the Fenestrata lace that you see attached. So Archimedes is a very, very famous and very common Mississippian bryozoan that we use to actually identify Mississippian-age rocks. Finally, order Cyclostomata, or round mouth, sometimes called Tubuloporata, is another well-known and modern bryozoan group. These are around from the early Ordovician to today. They have these long tube-like structures in their colonies that are unpartitioned, so they don't have those little floors and ceilings like we see in some of the other bryozoan groups. Their branches are very, very delicate, as you can see from the photograph of the modern bryozoan, the living cyclostomate bryozoan. And they're relatively rare in the Paleozoic, yet for some reason they are able to make it through all of these major mass extinctions and still persist today. This could be because they have a very simple form and they're living in areas that are highly buffered. So modern day bryozoans and modern day brachiopods are still part of our ecosystems, right? We still have a number of these around, and by studying them today, we can tease out some information of what some of our Paleozoic environments used to be like. So while these used to be much more abundant, both of these groups, in the Paleozoic, ever since the Permo-Triassic extinction, they've become rather minor. However, they're still around, and we can study them to give us a better picture of what those Paleozoic ecosystems looked like. So that concludes our Unit 7 or Week 7 material. We've covered sponges, archaeocyathans, cnidarians, and then all of the Lophophorates. We've looked at brachiopods and bryozoans. Next week in lecture, we'll be looking at arthropods, and in lab, we will also be looking at some arthropod fossils as well. So if you have any questions, please feel free to get in contact me. Otherwise, I will see you in lab.